Okay, now we're going to pick up at conduction of action potentials in your neurophysiology notes. We're going to start looking at what happens once an action potential is triggered at the axon hillock. And so let's go back to our lecture notes and look at some of the characteristics that we have. First, it's really important to remember that action potentials do not grow weaker as they travel down the axon. Therefore, they are called non-decremental. They do not grow weaker. And there are two kinds of conduction that are involved: electrotonic ion flow and regenerative ion flow. Electrotonic is what happens in the cell under the myelin. Do you remember how we have myelin sheaths that cover the axon of some neurons? Sodium ions come in, if you remember those gated channels, and the sodium comes in, kind of elbows its way until it reaches underneath, inside the axon, another gate, which will then, another sodium channel, which will then change configuration, open up, and allow more sodium to come in. So what is happening underneath the myelin is the electrotonic. It's got cable properties. Think of an electric cord carrying the electric down. There are no sodium channels under the myelin, and that is a great thing. We're not wasting extra materials like proteins and stuff, and it is faster than the regenerative ion flow, which is what occurs at the nodes of Ranvier, if we're looking at that area. The action potential gets re-triggered there. That node of Ranvier, if you need to look back at your early neuroanatomy notes, is where there will be another channel, sodium channel, that will open up and allow more sodium to come in. And so that is slower than electrotonic, because think about it. You have to open the channel, sodium comes in. So if you did that all the way down the axon, you would be taking a lot of time. But these channels, these at the nodes, are spaced, so we continue to get that regeneration of this action potential, this influx of sodium ions, depolarizing the axon, remember depolarization, until we get to the very end of the axon. So remember, it is slower, but it is more sure to make the action potential have the energy to get to the end of the axon. I kind of have the analogy in my head of swimming down an Olympic-sized pool, which would be kind of a challenge for me to start with. I'd like to go slowly, but I need to go fast, and I need to make it to the end. Well, if I dive off the end of the pool and I go underwater, which would be like electrotonic ion flow, I'm going, and I'm going pretty fast. But all of a sudden, it's like, I'm running out of energy. Oh my gosh, what am I going to do? I'm going to have to pop up to the surface, get some air, and that would be regenerative, and then go down and keep going. That way, I would have better success at reaching the end of the pool and still trying to do it quickly instead of swimming in some other, like a crawl, butterfly, which to me is like exhausting, breaststroke. So I'm going underwater, electrotonic, and then coming up to regenerated every so often. It's faster, makes it to the end. I get to the end of the swimming pool, which is a really good thing, because now I'm tired and just want to get out and rest. So if you put these two together, you end up with something that we call saltatory conduction. Saltaire means literally to dance. And if you put these two forms together, you then end up with this saltatory conduction. You get the best of both worlds. When I first learned neurophysiology back a number of years ago, I unfortunately had an instructor who was found it very hard to communicate certain concepts. And I could not figure out for the life of me the way she described it, that saltatory conduction was basically not a separate kind of conduction, which is what it kind of seemed like when I first learned about it. I sat down, thought about, looked it up, read books, and then realized that it is the combination of these two forms of ion flow. And again, the advantages are you don't have to have so many sodium potassium transporters, okay? Remember those sodium and potassium pumps. And you don't need so many, nor do they have to work so hard. And this conserves energy. Non-myelinated axons would be slow. For speed, the transmission between the nodes is very fast, and then getting regenerated to the nodes just make sure that this potential reaches the end. In some diseases, we have the degeneration of myelination, which means the signal travels more slowly down the axon. Here, I've included another link for you. And again, you can open this yourself, but let's do this for a moment. Here. And here you'll see this myelin, again, notes of Ranvier, right? This is showing a different way you can have the myelination. This would be like the oligodendrocyte that has the different processes, I think very like octopus-like, reaching out. This is unmyelinated, and so it's going to take a long time to get down here. It's going to take that potential, that change, a long time to move down because it doesn't have the opportunity to move fastly with electrotonic flow. So this is an example. Again, here is the myelinated axon. Here is where sodium channels would exist, transporters only here, not underneath the myelin. So hopefully that is a little bit useful. Again, here is unmyelinated. You see we don't have these myelin sheaths. So there's another link there for you. This, just want to make sure I open this so you see I'm not just filling this with stuff that isn't too meaningful. But this is more about the actual conduction, refractory periods. I want you to see all of the different things that are available here for you. Ion channel protein discussion, ions, the cations and anions, the sodium potassium pump. This, I feel, is really a good way of giving information without requiring a textbook. Here is measuring the resting membrane potential. Remember, we went through that. So factors that determine the resting membrane potential. So if you come back to this link, you are going to see everything from setting up that resting membrane potential to how the myelin helps the potential make it to the end quickly. Remember, saltatoric induction, combination of electrotonic, which is fast, regenerative, which occurs at the nodes, and make sure you make it. So I just want to make sure you remember this particular resource. I think it's very helpful and certainly will take the place of many textbooks. So now let's say we get to the end of the axon. And this is where I wish so much I was in the classroom with you, because it's so much, I think, more interesting when we can get up and actually act out these things. But unfortunately, that's not the case right now. So we're going to do the best we can. So synaptic transmission, when you get to the end, to the terminal button, there is a space between that terminal button and the next neurons that are in that area. And it's through this gap that we have the chemical transmission of signals. But how does that even happen? First, we need to synthesize. We need to build these neurotransmitter molecules. We need to package them, and we need to transport them. Now, lots of times we think of all neurotransmitters being synthesized only in the cell body, and that's not necessarily the truth, because small molecule neurotransmitters, which we'll be talking about in like the next segment of our neurophysiology material, are actually synthesized, or can be, in the buttons, that terminal end, fancy bouton, and packaged in little vesicles, like little baggies, if you will. And they're stored right next to that presynaptic membrane. So if you have your axon, this is your terminal button, okay? And here inside, you would have some of these vesicles waiting to just be released, okay? And how do they get there? Again, for the small molecule neurotransmitters, they have the ability to be synthesized right in the button. That differs from peptide neurotransmitter molecules, which are larger, and these are composed of 10 or fewer amino acids. There should be an F, and let me just change that right now. And these are synthesized in the cell body. And remember the microtubules? If some of these terms sound unfamiliar to you, go back and take a look at the very first lecture in neuroanatomy. In that lecture, you'll find all of these terms. You'll find the diagrams that you can take a look at. Those microtubules, like little shoots that'll take them right to the end, to the terminal buttons, then carry those molecules so they are available for release in vesicles. They're hiding out, they're hanging out, and then we'll see in a minute how they empty into that synapse, the synapses space between the one axon and the next neurons. Now, both small molecule neurotransmitters and peptide neurotransmitters can exist in the exact same neuron. Sometimes people get the idea, oh, there's only one in this neuron. No, because our body is so efficient and environmentally friendly that it can actually multi-purpose. Okay, now we need to get these neurotransmitters out into the synapse, or else they're never going to be able to go on and activate receptor sites on other neurons, or even on the same neuron that released it. And that's something we'll get to Hopefully you're like, wow, I didn't think about that being possible. Some of you do, because we've talked about another class if you've taken it with me, and some of you who've had bio classes probably are aware of that. But the process of releasing the neurotransmitter into the synapse is called exocytosis. And that should sound really like, oh yeah, exo, exit the cell, cyto, the process of exiting the cell, okay? So this is no mystery. Again, that's what we call this process, but how does it even happen? Okay, remember we have this action potential that comes down that axon non-decrementally. When it gets to the end, it will actually stimulate calcium channels. There are calcium channels at the end in the button and around that region of the neuron that are voltage activated. What's an action potential? Voltage. So we're going to open up those calcium channels. When those calcium channels open up, calcium ions enter. And when the calcium comes into the button, it causes those synaptic vessels, those little baggies of neurotransmitters, to fuse with that presynaptic membrane and empty their contents into the synaptic cleft, that space. In other words, here I am, I'm just like a happy little vesicle full of neurotransmitter molecules. Suddenly, and I'm going to say this here in front of me is the synaptic cleft, the synapse. And my computer, you can't see it, but imagine it around me. My computer screen is actually the membrane, presynaptic, because this is where we're releasing the neurotransmitter molecules. So here I am again, the little vesicle, calcium, whoa, whoa, whoa. I come and I merge right into this membrane, opening up myself and allowing all those neurotransmitter molecules that I've been holding on to be released into that synapse, that synaptic cleft. And so now I'm actually becoming, like, the vesicle part of me is becoming one with the membrane of the neuron from which I was released. Now, depending on what kind of neurotransmitter molecule I am, I can either inhibit the postsynaptic neuron, postsynaptic, the neuron that's after that synapse. I can say, no, don't, be quiet, be quiet, be quiet. Or I can excite it. It's like, go, get going, get going, get going, fire, fire, let's do something, let's do something. So I can either be inhibitory or I can be excitatory. So now we're moving on to activation of receptors by neurotransmitter molecules. Sorry, this is a little larger font, but hopefully this will still do the trick for you. So now here I am, a neurotransmitter molecule, and I've been released into that synapse. And there is a neuron there with receptors. Remember those proteins embedded into the cell membrane? Now, I can only influence cells that have a receptor for me. If I am GABA and there's no GABA receptors, I can't do anything. Okay, so there is specificity to some degree, and we will get to more of that later. In fact, we'll integrate some of that into the mental disorder biological mechanisms as we work through. But there are one neurotransmitter can bind to several different types of receptors, which are subtypes of that receptor subtypes. So you can have a class of receptors. And perhaps I can actually stick in so that I can activate it to any receptor subtype that is in that class. And this is possible depending on the neurotransmitter. Now, here is a term you're really going to need to know. It's simple, ligand. Ligand. Any molecule that binds to another is referred to as its ligand. Okay, you can probably figure out by now that that's going to be one of the terms you're going to need to know later on. Now, I'd like you to think back to when I was talking about the cell membrane, that phospholipid bilayer with two different types of protein receptors. Ion channel linked receptors is one. That's ionotropic. So this matches up ion. So I am trying to do a little demonstration to show this, and I need a different camera. So hopefully I can accomplish that and stick that up on the site as well. But hopefully this will make a little sense to you. Okay, in this kind of receptor, you have a channel. It's basically a channel. Now it's closed. Pretend this is the receptor and the interior of the cell is that way, okay, and it's closed. Now, all of a sudden, it's activated by a particular neurotransmitter. It opens up. Now, there is a channel going right through to the inside of the cell. Usually, this channel will open or close immediately, and that leads to a PSP. You can write in there post-synaptic potential. Post-synaptic potential. Now, a neurotransmitter may open sodium channels, and that would depolarize that neuron or excite it, right? If you depolarize, you're going to get action potential, or at least more likely to get one. Or this here's a different neurotransmitter. It may actually open a potassium channel, and if it does that, it's going to hyperpolarize, calm that neuron. It won't excite that neuron, okay? So again, this comes to excitatory and inhibitory. Now, these are fast. If you bind to one of these ionotropic receptors, the effect is fast, but it's also very short-lived. At any rate, these ion channels that open like I just demonstrated with my hands, kind of, sort of, are called ligand-activated ion channels. And here's a little illustration you can take a look at your leisure, and hopefully it will help. Now, I'm seeing all kinds of problems here. It's what happens sometimes when I'm trying really hard to get things done quickly to get them up for you. And I apologize, but okay, the other kind of receptor, the one that has one end that is outside of the cell, and again, I'm going to try to do a little demonstration for you that will show this a little bit more clearly, but we have an illustration coming as well. Just get this down: signal proteins and G protein linked receptors. That's the other kind of receptor, and they're called metabotropic. And I think that'll make sense in a minute. In this case, the neurotransmitter molecule attaches to a portion of the signal protein that is sticking up outside of the neuron in the extracellular fluid. There is a G protein. If you're a biology major, you know about G proteins. If you're not, just remember G protein for now. Is attached to a portion of that signal protein inside the neuron. So I just want to come down here for a minute. So we can kind of show this a little bit more readily. Here is the channel, ionotropic receptor. Do you see how we have the binding site, this opens, and now we have the influx of an ion? Whereas here we have a G protein or metabotropic receptor. I wanted to bring this up now, otherwise it's all going to sound like gobbledygook up above with the text. So here is that part of the signaling protein that I mentioned that's sticking out. This is extracellular. So let's say this is a neurotransmitter molecule I have released from my vesicle as I fuse into the membrane of the neuron from which I came. And here's a binding site. Now we're going to have parts of this go underneath. This is intracellular. This is within the cell. And here, oh, look at this, G protein. Great, just heard about that a minute ago. Now different things can happen. This is underneath, this is inside the postsynaptic neuron. This is inside the neuron that is being stimulated by the neurotransmitter that was just released. So let's go up for a minute and look at the things that can happen when you have a neurotransmitter molecule that binds to a metabotropic receptor, okay? Now, a subunit or a piece of the G protein will break away and do one of a couple of things, okay? This is inside, remember, inside the postsynaptic neuron. Neuron from which the neurotransmitter molecule comes, neuron that is postsynaptic that's going to be activated, the neurotransmitter molecule going that way. That piece might move along and bind to a nearby ion channel. Oh, wow. Just think about that. If I do that, if I move down, here I am, a little piece of G protein, and I go underneath and I see an ion channel, an ionotropic channel, I go and I bind to it, that'll open it up right away. So do you see they're cooperating? A metabotropic and ionotropic channel or receptors are cooperating. Or I could, as a G protein subunit, trigger the synthesis of a second messenger, which will diffuse through the cytoplasm. Remember, this is inside of the cell to which that neurotransmitter that has been released attaches, okay? So these are the two things that this G protein can do. Move along and bind to a nearby ion channel or trigger the synthesis of a second messenger, which then diffuses through the cytoplasm. Well, what will the second messenger do, you ask? Well, it can either bind to this ion channel. So instead of this exact G protein, it's a second messenger. It's like I sent my dog outside to take the plumber who was here yesterday a tool, okay? He's my messenger, my second messenger. I would be the first, I would be that G protein, but here you go, okay? Or I could influence as this second messenger, I could influence the metabolism of the cell. I could change metabolic processes inside of the cell. Well, I'll be darned, look at that. These are called metabotropic. Okay, makes sense. Or I could enter the nucleus. I am a tricky second messenger. And I can get right into the nucleus of this postsynaptic neuron and start messing with the products so that I am influencing the gene expression of that neuron. This is a big deal. Now, usually metabotropic activity is slower, but it is more diffuse and has varied effects, which are relatively long-term in contrast to those fast but short-lived effects of the ionotropic receptor. They are more prevalent than ionotropic receptors as well, okay? And we can be doing some review of that. So, neurotransmitters can also bind to receptors on the presynaptic membrane. Wait a minute. Here is the neuron that is releasing neurotransmitter molecules into the synapse. And here is the postsynaptic neuron that can be activated by these molecules. But guess what? There's a, here, that's a bit, I found out how poorly I sleep by having this on at night. Anyway, this right here is a receptor for the same neurotransmitter that is being released by its neuron. So now we have a neurotransmitter molecule that actually goes up and can bind to this particular receptor, this metabotropic receptor. Hey, it's on, you released me, and now I'm coming back and binding to a receptor right on you. I don't want to leave you. That's not really what they're thinking, but it kind of gives you a picture. Anyway, these are called autoreceptors. So this means that a neuron can bind its own neurotransmitter. The important thing about this is these autoreceptors can serve to monitor and decrease the release of that neurotransmitter. Okay, I'm going to take a break here. I think I will just post this as a separate video again, just so you have little chunks and you're not like caught up in one huge long one. Also, so I can kind of make sure that I'm taking you in the right direction at the right speed. Hopefully this is helpful. And so I'm going to end right here for now, but this lecture will continue in the next video coming up. I'm going to stop this share for a minute. And again, this is not the end of the neurophysiology lecture. I'm simply stopping here and using this segment for a video lecture. And then I will be doing the last section in a separate one. They will all fit together, all probably be 3 separate little video lectures, all having to do with neurophysiology. But hopefully just breaking it up helps make it a little bit more manageable, and hopefully you'll be able to kind of, oh, I need to go back and refresh my memory about something. It won't be so hard to find. So there is rationale behind my madness sometimes, and hopefully this will help, and I will see you soon in the next video lecture.